So I wanted to see if I could give you guys a little bit of help with the muscle tissue lecture that we missed on Monday due to the snowstorm. So I'm going to try to teach using some simple things like a piece of paper and some markers in my own voice to see if I can help you out a little bit and try to explain some things. So where this topic picks up is with Roman numeral 5, the behavior of muscles. So we're going to be looking at how it is that muscles actually accomplish that task of shortening and causing, in most cases, movement of the skeleton. So we pick up with letter A, the topic of excitation, and what this is about is to explain how it is that that motor neuron is able to effectively transfer its excitement of electricity to the muscle cell to get it to become electrically active as well. So last week we talked about the features of the neuromuscular junction, and so today this topic is about putting it together and seeing how it works. So getting back to where we were, we had this idea of the end of a motor neuron flaring out to become that synaptic knob, again, which we talked about last week. Some other things of importance here to remember that we talked about would be the presence of calcium ions in high quantities outside of the motor neuron in the extracellular fluid. Remember how they got that way? How do we set this up? Is that we have calcium pumps in the wall of the motor neuron, especially here at the end. And those pumps are always using ATP to transfer and actively transport calcium ions out to the ECF. At the same time, we also need to have, as we'll see in a second, the opportunity to allow the calcium ions to come into the motor neuron at the right time. So these would represent then calcium gates, which can be closed or opened and allow the passive diffusion of ions of calcium in or out, whatever they want to do. And then these represent the calcium pumps that use ATP to constantly send calcium ions out. So to start the process off, we find that the brain has decided to send electrical signals down the motor neuron to try to stimulate the muscle cells of its motor unit. And so we might say our first step here is to see electrical signals traveling down, ends up traveling down the plasma membrane or the surface of the motor neuron. So that's step one, would be to see the brain accomplishing that. Step two then tells us that when electricity is present in the membrane of the motor neuron and it reaches this synaptic knob area, that will cause the calcium gates here to change to the open conformation. And that means then that calcium ions, despite the pumps, will flush into the synaptic knob or the motor neuron itself. So that would represent sort of step two of the excitation process, is that the presence of electricity down the motor neuron stimulates calcium gates to open and calcium ions diffuse in. Okay? Step three then says that the presence of calcium ions inside of the motor neuron, so here they are approaching the synaptic knob, the very end there. The presence of calcium ions inside has an effect on the synaptic vesicles that are housed here and contain inside of them the neurotransmitter called acetylcholine. So these large bubbles are the synaptic vesicles, and the little light blue dots that I'm placing inside represent acetylcholine inside of here. When calcium ions are present in here, and remember normally they're not, the gates are normally closed, the pumps keep them out. But when they're triggered to come in and they reach the synaptic knob, that causes the synaptic vesicles to rush to the end of the motor neuron, of the synaptic knob here, and release their acetylcholine into the synaptic cleft. So they release a chemical into that space just in front of this motor neuron. And remember, there's an object not too far away, but at a distance that we're trying to excite. This represents then the sarcolemma of the muscle cell. So the third step would be to see the release of acetylcholine into the cleft. The fourth step would then be to say that with an abundance of acetylcholine suddenly on this end of this watery cleft, we would expect then the acetylcholine to rush across to an area where it is deficient, just due to simple diffusion, to the sarcolemma side of the cleft. So that would be our step 4, would be to see acetylcholine diffuse across the cleft to the sarcolemma side. And step 5 says that waiting for it over there, and what it binds to, are specially shaped receptors on the muscle cell surface, acetylcholine receptors. And they bind to that. They have an affinity for those receptors. And the final step is to say that when that happens, the presence of acetylcholine in the receptors causes the muscle cell's sarcolemma to develop its own excitement wave and radiate that out across its surface in all directions. So step 6 is to finally reach that accomplishment of excitation, of saying now the sarcolemma is excited. It took these steps to get to that point. Brain sends electricity down the motor neuron, causing calcium gates to open. Calcium diffuses in, and when it reaches the knob, it causes synaptic vesicles to release acetylcholine into the cleft, which diffuses rapidly across to the other side of the cleft, where it binds with acetylcholine receptors on the sarcolemma, and that triggers new waves of electricity to go across the surface of the muscle cell. So now it is excited. So the challenge was, how do we get electrical excitement in this cell to transfer to this one? The issue was the cleft. There is a gap here. So the electricity cannot physically transfer from one cell to the other because they're not touching. So what we needed to do was change the message from an electrical one to a chemical message and back to electrical again. So the acetylcholine, that's what neurotransmitters do. The acetylcholine was our go-between, between the two cells, to help us physically cross that cleft with a message of some type to get the message to continue into the other cell. So that's the story of excitation, how we go from a neuron being excited to a muscle cell being excited. The next process that we look at is how do we then expect that excitement of the muscle cell at its surface, the sarcolemma, to have an effect down at the myofilament level, the thick and thin filaments. They're going to eventually need to cross over each other and slide. But right now we're not really anywhere near that. So we need to think about how we get that message down there. So this would be letter B, the excitation to contraction coupling story. Right? So that's where we go next. Excitation to contraction coupling. So let's represent here, let's pick up our story from the sarcolemma. So here we'll just draw a great big sense of imagining a fibrous muscle cell. And the red line is its plasma membrane, its sarcolemma. And then let's add a few things here. Remember, we have these tunnels that allow the sarcolemma to coat the lining of those, and so whatever's happening on the sarcolemma can technically go down these tubes to the other side without being released inside of the cell. And then what else is important is to remember the smooth ER, or the sarcoplasmic reticulum of the muscle cells, is deliberately allowed to pass very close to those transverse tubules, or the T tubules. Right? So what I've drawn here in blue, this is the sarcoplasmic reticulum. And once again, we have a familiar participant here. We have calcium ions that are stored inside of the sarcoplasmic reticulum. And that always then assumes we're going to need to see something that allows them to get out of the sarcoplasmic reticulum, calcium gates. And then we also need to explain and remember how the calcium ions got so abundant inside there, what trapped them in there, and that would be the role of the calcium pumps. So remember, those are always putting calcium in, and as long as the gates are closed, the calcium stays in. But if you open the gates, they're going to come rushing out, and as you already probably know, we're going to do that. So how do we get to that point? Let's pick up where we left off from excitation, and we saw waves of electricity from that acetylcholine receptor bonding radiating along the length like this. So that's a repeat of step 6 from the last process. Where we pick up this story is say, yeah, then what? So what we say happens, what we know happens, is that the electrical signals, because of the transverse tubules, will be able to travel down through the core of the cell, but again, without being loose in the cell. So it's still contained in an outside environment, but it can be experienced deep inside of the cell. So that would be, if we wanted to continue the numbering, that's fine. We could start over with numbering. It doesn't matter to me. But here we would say then, let's continue. Here would be the 7th step of muscle contraction, saying that the electrical signal from the sarcolemma travels down the transverse tubule. When that happens, its nearness to the SR causes, just like we saw in the motor neuron, causes the calcium gates that are normally closed to open. So nearby electrical activity triggers calcium gates to change to the open conformation. And so we would then see calcium ions exiting the sarcoplasmic reticulum. So we might say that represents a visual of the 8th step in this process. So the release of calcium, and what does that do? What's the benefit there? So what that's going to lead to, this isn't to scale or anything, but where we're headed is to those thick and thin filaments. And so if we have representation of, just really quickly here, a thick filament with all those myosin heads, just they're sitting out here in stacks, just out here outside of the SR. So I'll just draw one representative one there. And then here's an example of one of the thin filaments, right? Then the last thing to mention is that the calcium ions really like the troponin on the thin filament. So we would say at this point that the calcium ions will then go and approach the thin filaments looking for troponin. So if we can bring the story from the sarcolemma to an idea of we are now bringing the story down to the thin filament location, then we're shrinking even smaller, but we're getting closer to the actual contraction of the muscle cell. So that's the excitation-contraction coupling story that brings us to the thick and thin filaments. So then the third component and final one of contraction is the sliding filament theory. And remember from chapter 1, a theory is not a guess in science. It's something that's proven, and all we're doing is saying, by calling it a theory, is we're just holding back from calling it a law. So we definitely know that this happens, and we're just continuing to add to our knowledge of this process. But we're not questioning that it happens. Okay. So in the sliding filament theory, let's get back to, this one's a little harder for me to draw. Things change a lot, so we'll see how this goes. But here we come back to the idea of a thick filament with all of those myosins and their golf club heads protruding out in all different directions. And then we had the thin filament composed of those actins in a chain-like fashion. Right, I'll just do one side of it here for the sake of time. And so we've got tropomyosin is resting frustratingly for the thick filament over the special binding sites of actin. And then we have the troponin snowman every once in a while. Right? So there's our thin filament with its 3 parts to it. So we pick up the story here by remembering step 9 was that the calcium ions that got released from the SR, they're going to come to troponin, and they have an affinity for each other. So they bond, and calcium bonds to troponin. So since we didn't explicitly say it does bond to troponin, we'll go ahead and say that's our 10th step in this process. Troponin binding, or excuse me, calcium binding to troponin. Okay. So then what happens? Well, I'll come down here and let's think of the thin filament a little bit larger, hoping you can see this. Remember that the actins along the chain, all of these proteins, have a special binding site that the myosin heads really enjoy bonding with. And the reason they're not doing it currently is that tropomyosin is resting across them. So if we bring in just one troponin here, when calcium, I'll put a little green dot there, when calcium binds to troponin, it triggers troponin, the little snowman, to roll to another part of the actin string and bring tropomyosin with it. So step 11 is to say that troponin causes tropomyosin to shift off of the binding sites on the actins. So here are those binding sites. And tropomyosin, my green string here, is now somewhere else. It's on a different part of those red balls that I've drawn. So here's troponin. It did this. And then the calcium is still bound to it, causing it to move like that. So this would represent sort of step 11. Troponin moves tropomyosin off of the actin binding sites. Okay? And then what happens is that the myosins of the nearby thick filaments can now use ATP to spring up and attach to those binding sites. So I'm going to leave tropomyosin and troponin off of this picture. We don't really need them. But if I can take one of these and show you, here's a myosin that came up, right, from the thick filament, sprung up here at the hinge section, and attached itself to wherever it could on one of these actins, on the binding site there. So that would be a 12th step to say myosin uses, we can put it here, uses ATP, our energy molecule, to reach up and attach itself to actin and give a pull inward. These thick filaments and their myosins, they take, if I can use my hands here, see if this works, but if the actins are facing each other like this, the thin filaments, and there's a gap between them, then the myosins will reach up and give a little tug, and the ones over here will reach up and give a tug. And so what they're trying to do is use ATP to pull the thin filaments so that they touch in the middle. When left alone, they have a gap, but we're trying to bring them together to meet in the middle. And it's the myosins that do that. So back up here on this picture with the thick filament, these myosins, if they're allowed to, will attach to and pull this thin filament this way. And I didn't draw it, but there's another thin filament over here. These myosins will attach and pull this thin filament this way. So we could say this represents maybe the idea of our 12th and maybe our 13th steps. 12 you could say is attaching using ATP, and 13 is then to give the power stroke. That's what we call that little shift of the heads of the myosins when they do this. That's called the power stroke, and it pulls those thin filaments. They keep doing it. They let go and need a fresh ATP to get a fresh grip. But while a certain myosin has given a tug and needs to recharge itself again to regrip, other myosins are doing the same thing. And so you get this hand over hand sort of an idea of pulling the actin subunits, the thin filaments, together into the middle. So this kind of ends the story then. So we see the filaments, maybe I'll do it this way. we see the filaments then, the thicks stay in one place, but they move the thin filaments across like this. Or if I even do a better job like this, then here's two thin filaments, the thick filament controls them, uses ATP to bring them together like this. Well, if you do that, remember the concept of a sarcomere, the edges that all these things are attached to, those Z discs, will be drawn in with them as the filaments slide across each other. And so the last step, you could say the 14th, is that with the successful sliding of the myofilaments, the sarcomere itself shortens from side to side, from one end to the other, is brought closer together. And every sarcomere is going to experience this within the myofibrils, within the muscle cell. And so if every sarcomere shortens, the entire cell can then be expected to shorten and pull with some force on whatever it is that muscle is trying to do, on the bones and whatever they're holding, so to lift a weight or something. So that's how contraction works, all the way back to the excitement of the motor neuron, eventually trying to get the filaments to slide and the sarcomeres to shorten.